This video is about RNA editing and mRNA transport. Let's learn about RNA editing. RNA editing is a process of RNA modification in which discrete changes are made in the specific nucleotide sequences within an RNA molecule after transcription without splicing. In a simple way, it can be defined as any type of changes in RNA transcript sequence without RNA splicing called RNA editing. It was previously thought as rare phenomenon, but it is widespread. It was firstly discovered in mitochondria of trypanosomas. Just remember the sleeping sickness caused by this trypanosoma. So with that, just remember it was firstly discovered in mitochondria of trypanosomas. Trypanosomes. It takes place in cell nucleus and cytosol as well as mitochondria and plastids. RNA editing enhances a cell to obtain large diversity in proteins by using small number of genes. It is mainly achieved by insertion, modification or deletion of one or more bases. It happens after transcription, also happens in poly A tail. The coding sequence of an mRNA molecule is already the coding sequence of an mRNA molecule is altered after transcription in mRNA editing so that the protein has an amino acid sequence that varies from that of the gene encoded. RNA editing describes the alteration of an RNA sequence by introducing or removing nucleotides from an RNA or by changing the character of a nucleobase by deamination. For therapeutic purposes, the two types of deamination reactions, converting cysteine to uridine and adenosine to eosine, are considered most useful. RNA editing occurs in the nucleus as well as in mitochondria and plastids, which are thought to have evolved from prokaryotic-like endosymbionts. There were substitutions in some of the mRNA nucleotides in some molecular genes, in nuclear genes in mammalian cells and in some mitochondrial genes in plant cells. More exclusive RNA editing for certain mitochondrial genes in trypanosomes, parasites, has been found in the mRNA. More than 60% of the sequence is determined by RNA editing in some of these organisms' mRNAs. In RNA sequences, a variety of mechanisms can bring about changes. Molecules called guide RNAs, gRNAs, play a key role in certain situations. Guide RNAs or gRNAs consist of sequences that are partly complementary to pre-edited RNA segments. Guide RNAs or gRNAs consist of sequences that are partly complementary to pre-edited RNA segments. In these sequences, the two molecules goes through base pairing. The mRNA undergoes cleavage after the mRNA is anchored to the gRNA and nucleotides are inserted, removed or altered according to the gRNA template given. The mechanism of RNA editing can be divided into two categories, addition and deletion or substitution. So types of RNA editing. RNA editing is mainly of two types. First, base modification, site-specific deamination of cytosine and adenine, A to I, C to U, C, etc. Enzyme deaminase is involved. Insertion and or deletion, guide RNA-mediated site-specific insertion and deletion of uridine base U, insertion, deletion, and so on. Seen in kinetoplastid protozoa. Mono or dinucleotide insertion seen in Physarum. Nucleotide replacement seen in Acanthomoeba tRNAs. Then addition and deletion. The first way that RNA can be edited is through addition. In addition editing, new nucleotides are inserted into the original sequence. Then substitution. Another type of RNA editing is substitution. Enzymes bring about the conversion of the base in other cases. For example, in humans, a gene is transcribed into mRNA that encodes a lipid transporting polypeptide called apolipoprotein B100, which is synthesized in liver cells and has 4,5,6,3 amino acids. By editing the apolipoprotein B100 mRNA, a truncated version of the protein called apolipoprotein B48 with only 2,153 amino acids is synthesized in intestinal cells. A cytosine base is deaminated by an enzyme in this editing, transforming it into uracil. This conversion converts a codon that specifies the glutamine amino acid into a stop codon that terminates translation prematurely, resulting in the protein being shortened. Then base modification, apolipoprotein B gene, codon 1 and codon 2 liver, unedited mRNA, translation, transcription, intestine, unedited mRNA is their translation, lipoprotein in transport of cholesterol in the blood, function absorption of lipids from the intestine. Base modification, deamination of adenine and leucine. Deamination of adenine and leucine E to I modification usually occurs in secondary structure regions of RNA. Set of protein required for deamination of adenosine called DAR or dsRNA dependent. This leucine can be base paired with either A, T, G or C. Leucine is recognized as guanine during translation. Example includes different types of GLUT receptors for insulin, adenosine, leucine and guanosine. And deamination. The 8I deaminase enzyme DARs. DAR stands for adenosine deaminase acting on double-stranded RNA. This enzyme catalyzes the hydraulic deamination of adenosine to leucine in double-stranded RNA. Leucine undergoes non-canonical base pairing with cytidine, which is and is interpreted by the translational machinery as a guanosine. So insertion or deletion. RNA editing through the addition and deletion of uracil has been found in kinetoplasts from the mitochondria of Trypanosoma brucei. It is sometimes in a large fraction of sites in the gene, therefore it is also called the pan-editing, so distinguish it from typical editing of one or a few sites. It starts with the base pairing of the unedited primary transcript with a guide RNA or gRNA, which contains complementary sequences to the regions around the insertion or deletion points. The newly formed double-stranded region is then enveloped by an editosome, a large multi-protein complex that catalyzes the editing. The editosome opens the transcript at the first mismatched nucleotide and starts inserting uridine. The inserted uridine base pairs with the guide RNA and insertion continues as long as A or G is present in the guide RNA and stops when a C or U is encountered. The insert nucleotide then causes a frame shift and results in a translated protein that differs from its gene. The mechanism involves an endonucleolytic cut at the mismatch point between the guide RNA and the unedited transcript by editosome. Then insertion or deletion. Then uridine are added from UTP at the 3' end of the mRNA by one of the enzyme of the complex called terminal U transferase, and paired U's are removed by another set of enzymes known as U-specific exoribonuclease. Editing leads to the formation of mRNA complementary to gRNA, which then rejoined at the ends of the edited mRNA transcripts by RNA ligase. As a consequence, the editosome can edit only in 3' or 5' direction along with the primary RNA transcripts. So the complex can act on only a single guide RNA at a time. Therefore, RNA transcript requiring extensive editing needs more than one guide RNA and editosomes complex. So addition of uracil to RNA. DNA is there, pre-edited RNA, transcription of DNA to pre-edited RNA, then gRNA. Guide RNA gives template for editing. Then pre-edited RNA, editing of pre-mRNA. Then translation to protein or other pathway. Guide RNAs or gRNAs, direct editing. gRNAs are small 40 to 70 nt and complementary to portions of mRNA. Structural elements anchor informational part and oligo tail, U-tail. Base pairing of gRNA with unaided RNA gives mismatched regions which are recognized by the editing machinery. Machinery includes an endonuclease, a terminal uridyl transferase, TUTase, and RNA ligase. Editing is directional from 3' to 5'. Addition site, deletion site. Then RNA editing mechanism. Editing is catalyzed by a multi-protein complex that have not been fully refined yet. It contains following four key enzymes as characterized from glycerol gradients of 20S editosomes. Endonuclease cleaving in vitro occurs in an unpaired nucleotide immediately upstream of gRNA and mRNA anchor duplex. Exonuclease it removes non-base paired U nucleotides after cleavage of deletion editing sites. Terminal uridyl transferase TUTAs it adds us to the 3' end of 5' mRNA fragment as specified by the gRNA. RNA2 ligase, the natural editing ligase substrates are nicked dsRNAs that are completely base paired after the correct addition or removal of nucleotides. Then helicase, it provides ATP dependent unwinding activity in RNA editing. Thus, it is involved in gRNA displacement directly from gRNA edited mRNA duplex or indirectly to allow 5' gRNA to form an anchor duplex with an edited mRNA to initiate another block of editing. Examples include REH1, RNA editing helicase 1 in trypanosoma, other 20S editosome proteins, RNA editing mechanism, involvement of enzymes in RNA editing, nuclease, TUTase or 3' exonuclease, ligase, then deletion or insertion, TUTase or terminal uridyl transferase adds US to the 3' end created by cleavage of the pre-mRNA. So, disruption of the RNA editing balance, locomotion, depression, schizophrenia, neuronal disorders and behaviors, PWS, and energy metabolism, obesity, MADAR2, GRIA2, cancer, leukemia, prostate, breast, lung, kidney, prostate, testis, glioblastoma, pediatric astrocytoma, leukemia, glioblastoma, lung, kidney, prostate, testis, breast, prostate, leukemia cancers. Then inflammation or interferon, systemic inflammation, autoimmune lupus disease, HDV, HIV, enhanced replication, hypermutation, VSC, measles, influenza, virus infection, disease group editing deficiency, hyperediting, misleading, genetic model, disruptions of DNA editing balance. Then CRISPR, clustered, regularly interspaced, short palindromic repeats. CRISPR is a powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. It has many potential applications, including correcting genetic defects, treating and preventing the spread of diseases, and improving the growth and resilience of growth. However, it has some ethical concern. It utilizes CRISPR-associated 9-Cas9, which is an enzyme that acts like a pair of molecular scissors capable of cutting strands of DNA. Thus, it is an RNA-guided gene editing mechanism called CRISPR-Cas9. It was adapted from the natural defense mechanism of bacteria and archaea. These organisms use CRISPR-derived RNA, a molecular cousin to DNA, and various Cas proteins to foil attacks by viruses. What is the significance of RNA editing? It is essential in regulating gene expression of organisms. RNA editing mutant has reported in strong effects in organelle development. Defects in RNA editing cause diseases. It is a mechanism to decrease the number of different proteins available without the need to increase the number of genes in the genome. So, defects in RNA editing cause diseases. Number of different proteins will be increased without the need to increase the number of genes in the genome. It may help protect the genome against some viruses. Next, how is RNA transported out of the nucleus? The process of nuclear export is similar to doing the opposite of nuclear import. During nuclear export, the exportin binds the cargo and RAN GTP, RAN RAS related nuclear protein, in the nucleus before diffusing through the pore into the cytoplasm. In the cytoplasm, the complex then dissociates. The resultant RAN-GDP complex is returned to the nucleus where it performs the process of exchanging the binding ligand for GTP. RAN-GTP is then able to connect to GAP and hydrolyze GTP. Therefore, whereas importance depend on RAN-GTP to dissociate from their cargo, exportins require RAN-GTP in order to bind to their cargo. This is because exportins bind RAN-GTP to their cargo. After the post-transcriptional modification process is finished, mature mRNA is transported to the cytoplasm by a protein that is specifically designed to act as an mRNA exporter. Although the particular mechanism behind this translocation process is not yet fully understood, this process is actively dependent on the RANT protein. Some genes that are transcribed more frequently than others are physically situated close to nuclear pores in order to make the process of translocation easier. The export of tRNA is likewise dependent on the numerous alterations that it goes through. As a result, the export of tRNA that does not function properly is prevented. This quality control system is essential because of the crucial role that tRNA plays in the translation process. During this process, tRNA is responsible for adding amino acids to peptide chain that is expanding. Exportin 1 T is the name given to the tRNA exporter found in vertebrates. The presence of RAN-GTP helps to speed up the process by which exportin 1 T connects directly to its tRNA payload that is located in the nucleus. tRNA's capacity to attach to export T as a consequence to be exported is inhibited when mutations that change the structure of tRNA occur. This provides the cell with an additional quality control step. So pre-mRNP, hnRNP proteins, CBC, mRNA, M7G, TAP, ALY, and IPB beta like export receptors, RAN, GTP, nucleus cytoplasm, NPC, mRNA export